Hi class. In today's lesson, we'll go through the methods that people have used to identify a genetic basis for human disease. How is a particular gene implicated for a particular disease? Initially, we had allocated 2 lectures for this topic, but the topic itself is quite complex, and I don't believe that you need to go through that in that much detail. As medical students, it's enough for you to understand the basics of how this is done. And if anybody wants to have more information, you can obviously go back to the textbook and read that in more detail. But most people, even clinical geneticists, are not too involved in identifying the genetic basis of human disease. How is a particular gene implicated in a particular disease? But usually it is scientists who do gene discovery who are quite deeply involved in this methodology. However, it is very, very useful for you to know a little bit, a background of how is a particular gene associated with a particular disease. How do we know that BRCA1, for instance, is associated with breast cancer? How have people identified that? So let's begin by just going through a little bit of background. So because we've combined these 2 lectures into one, you won't have a class. The next class would be a free hour. However, please utilize that time to go through the material that we've covered so far and identify any questions that you might want to ask or any doubts that you have. So how are genes implicated in disease? In order to identify which genes are associated with disease, there are 3 main approaches that have been used traditionally. The first and the most used and the oldest methodology that has been used is called linkage analysis. Linkage analysis is basically studying the co-inheritance in large family pedigrees of 2 linked markers. We'll go through that in detail in a little while. And linkage analysis is useful for monogenic disorders, single gene disorders. Genome-wide association studies is another way of trying to find what sort of genes or DNA markers are associated with a particular disease. It is studying the frequency of an allele or a set of alleles in large population-based cohorts. It is useful for multifactorial diseases. So we've learned about multifactorial diseases and monogenic disorders. One example of a monogenic disorder is cystic fibrosis or breast cancer caused by BRCA1 or BRCA2 gene disorders, not breast cancer, which is caused by polygenic causes, but monogenic, single gene type of breast cancers or cystic fibrosis. So those sorts of conditions are monogenic, whereas polygenic or multifactorial diseases, the examples include old onset dementia, for instance, or diabetes. Those are the type of conditions which are multifactorial. So a little bit of gene and a little bit of environment all mixed together increases a person's chance of having that disease. And genome-wide association studies can identify the genes which increase the risk of having that particular disorder. Another way, which is more recent, is direct genome-wide sequencing. So you can use whole genome studies or whole exome studies. So actually sequencing all of the genes or all of the DNA material and then trying to filter out the things that are not considered to be associated. And we'll go through all of these 3 in a little bit more detail in our next few slides. So what is linkage analysis? Before we start studying about linkage analysis, it's important to know something about homologous recombination in meiosis. So we know that in meiosis 1, so there are 2 homologs that pair with each other. Homologs are similar chromosomes, one that comes from the mother and the other that comes from the father, as you can see here. So there are 2 homologs that pair up. And then there's recombination that occurs in multiple parts within that chromosome. That creates like a patchwork effect in each of these chromosomes because it sort of exchanges genetic material from between those 2 chromosomes. And then when you think about the child of that person, they once again have had a lot of homologous recombination. So there is a lot more difference in the genetic material that has been inherited. So each chromosome inherited by a child from a parent is never exactly the same as either of the 2 copies of that chromosome in the parent. So this is the concept of human genetic individuality. So there is a lot of homologous recombination that occurs and exchange of genetic material, and this has been exploited and used in trying to identify which segment of all of these various colors you can see in the third generation, if you can narrow down a particular disease to just one segment, then you don't have to then look through the entire area of all of the chromosome to identify a particular disease. So using this concept of recombination narrows down the area of search for a particular disease, and we'll go through that in more detail. Another thing also to know is after homologous recombination occurs, so the patchwork effect, the gametes are independently assorted. So there are multiple possibilities of how the gametes are distributed in the child. So as you can see here, there are different combinations. So the uppercase A can then be inherited with uppercase B, lowercase a and lowercase b, or uppercase a and lowercase b, or lowercase a and uppercase b. So all of these combinations are possible. This is called independent assortment. So using a combination of homologous recombination and the independent assortment, we can further narrow down the segment of the chromosome or the DNA that we are interested in for a particular disease. So let's talk a little bit about the linkage analysis itself. So as we talked about earlier, recombinant is where there has been a crossover or recombination that has occurred. Non-recombinant is where there is no crossover. So let's consider the example of a few loci here. So you can see here in example A that there is a chromosome with locus 1, which consists of A, and locus 2, which consists of B. So there are two varieties in this locus, two genotypes, so uppercase A and lowercase a, uppercase B and lowercase b. Now, when the child inherits the uppercase A and uppercase B always together, then it is non-recombinant. If there is lowercase a and lowercase b always together, that is also non-recombinant. But if there has been a crossover between these two, and a child inherits an uppercase a along with the lowercase b, or vice versa, which is lowercase a along with a and uppercase b, then you know that there has been a recombination. Now, recombination, if you apply common sense, you would also know that if there is more distance between two of these loci, then there is a higher chance of recombination occurring. If these two loci, as in the second example, are very close together, then it is likely that they will always be linked, or they'll occur together, and that's likely to produce non-recombinance only. But if there is a reasonable distance between those two loci, then there is a possibility that there could be non-recombinance as well as recombinance. If there is a large distance between these two loci, then it is possible that there is always a recombination that occurs between these two loci. So how do we detect recombination events? So in order to detect recombination events, in order to know if there has been a recombination, there needs to be heterozygosity between the two chromosomes. So one has to be an uppercase A and another has to be a lowercase a. Otherwise, it is hard for us to know whether the crossover has occurred or not. If, say, for instance, both the genotypes are lowercase a, then we don't know which lowercase a is present in one chromosome in the child, whether it is from the mother or whether it is from the father, because we can only look at it as being either uppercase or lowercase. So detecting recombination events requires heterozygosity, and also we need to know the knowledge of phase. We need to know that A and B are a haplotype, which means that they are in the same chromosome, generally speaking, and if recombination occurs, then it can change. So we need to know the haplotype, as well as there needs to be heterozygosity in order to detect recombination events. So let's consider an example here. So let's say, in this example, in this pedigree, take generation 1. So you can see the mother here. The mother here has a blindness. We don't know what is causing the blindness, but we know that her children have also got the same type of blindness, some of the children. So there is a genetic component. There is something, a particular locus, a particular area within her DNA, which is causing this blindness. So we've narrowed it down. We know that there is this one particular chromosome. But there are 3 loci, and we don't know which locus, which one of these is actually causing the blindness. The blindness is due to retinitis pigmentosa. So all of her children, as well as her, have got retinitis pigmentosa. Not all of her children, 3 of her children, as well as her, have got retinitis pigmentosa. So what we need to do here is we know these 3 markers. We are able to sequence these 3 markers. right? So we've sequenced these 3 markers in the mother. So the mother's got in one chromosome uppercase B, uppercase D, and uppercase A. In the other chromosome lowercase b, lowercase d, and lowercase a. And the father has got lowercase b, lowercase d, and uppercase A, and lowercase b, lowercase d, and uppercase A. All right. So now we start sequencing each of the children. Let's consider the first child. So generation 2, person 1. So this person does not have blindness, so does not have retinitis pigmentosa, and they have a lowercase b, they have a lowercase d, and a lowercase a. So we know that lowercase b, lowercase d, and lowercase a are not associated with retinitis pigmentosa. We can exclude those 3 now. So let's move to person 2, the second child, who does have retinitis pigmentosa. So this person has an uppercase b, an uppercase d, and a lowercase a. We already know that a lowercase a does not cause retinitis pigmentosa. So it's got to be either uppercase b or uppercase d. So let's move on to the next person. So the third person also has retinitis pigmentosa. They've got a lowercase b, an uppercase d, and an uppercase a. We already know that lowercase b does not cause retinitis pigmentosa. And we know that the person 2 had retinitis pigmentosa and had uppercase D. So therefore we conclude that uppercase D is most likely associated with retinitis pigmentosa. However, just having these markers is insufficient, and it has to be statistically significant. So we need large pedigrees. We need to see this pattern in a number of people across multiple generations, and we generate something called a LOD score, which we'll talk about as well. And if there is statistical significance, then we know definitely that this locus is what is actually causing retinitis pigmentosa. And then this is how RP9, the gene RP9, was discovered. This area was then sequenced more thoroughly, and they found that there were genetic markers. So there is a start codon and a stop codon. That means that there is actually a gene, and that gene was indeed associated with retinitis pigmentosa. So let's take another example for disease-associated loci. So let's say in here, there is a large pedigree that is available to us, but we don't have the informative markers in everybody. But as you can see in generation 1, they passed away. We don't have their DNA. Generation 2, we don't have their DNA either. But this is still informative because we know here that there are 2 people that have inherited that. And when we see the pattern, it seems like it is autosomal dominant. So there is male-to-male transmission. So it's informative for us to know that this is a Mendelian disease. And then we've been able to sequence, or we've known that there are markers A, B, and C. So we can see that this particular person in generation 3 has got markers A and markers B, and is affected. And the person next to them is their cousin is also uppercase A and uppercase B. So AB and AB. So one of these 2 are possibly associated. And then we see further markers down here. So AB once again is associated, AB once again is associated in the next generation as well, in all of the people. And then we see in the generation after that AC is associated. So now we conclude that A is actually the locus, which is associated with this condition. So C is not associated with the disease because B is not associated with the disease because the ones who have inherited A is a common factor between all of the ones who have actually got the disease. So that's a very basic concept of how linkage works. So linkage and recombination frequency, going into a little bit more detail about how it is actually done and statistically done. So we've talked about the concept. This is the practical way of doing it. You don't need to know every aspect of this, but I'm going to try and summarize it and simplify it as much as I can. So linkage is a term used to describe a departure from the independent assortment. So we've talked about linkage disequilibrium, and linkage equilibrium in our previous slides. So linkage is a term which is used to describe departure from independent assortment. So we'd expect independent assortment most of the time, but when it is linked, then there is no independent assortment between the 2 loci. Or in other words, the tendency for alleles at loci that are close together on the same chromosome to be transmitted together as an intact unit through meiosis. Analysis of linkage depends on determining the frequency of recombination as a measure of how close two loci are to each other on a chromosome. A common notation for recombination frequency. So recombination frequency is a proportion, it's not necessarily percentage. So the proportion is, and the notation that's generally used is the Greek letter theta. So theta can vary from 0, and there is no recombination at all, to 0.5 when it is complete independent assortment. If two loci are so close together, the theta is 0 between them, they're said to be completely linked. If they're so far apart that theta is always 0.5, they're assorting independently and are unlinked. In between these two extremes are various degrees of linkage. Map distance is another terminology which is used. It may not necessarily be too important for you, but to see how distant those two loci are, it's depend on how many linkage, recombination events that have been observed between those two loci. It is measured in units called centimorgans, and distant alleles are those which are independently assorting, in other words, theta of 0.5. Closer alleles have theta of less than 0.5. Linked alleles, alleles that always occur together, have a theta of 0. So there's also linkage disequilibrium clusters. So clusters of loci with alleles in high linkage disequilibrium across segments of only a few kilobase pairs to a few dozen kilobase pairs are termed linkage disequilibrium blocks. So these blocks always seem to occur together. So these haplotypes occur together in everybody, and for some reason there isn't much recombination, either because they're closely linked or there might be other reasons as to why there isn't enough recombination between these. So as you can see here, there's a cluster 1 and a cluster 2, and these always seem to be occurring together. So you can see the haplotype frequency here. The haplotype frequency is about 98%. They seem to be occurring together mostly. Mapping human disease genes by linkage analysis. So it's how do we determine whether two loci are linked? Linkage analysis is a method of mapping genes that uses studies of recombination in families to determine whether two genes show linkage when passed on from one generation to the next. So the steps is first to determine theta, as we'd seen previously, then see if it is statistically significant using likelihood ratio. So likelihood of the data if loci are linked at distance theta divided by likelihood of the data if loci are unlinked. The computed likelihood ratio for different values of theta is usually expressed as the log 10 and is called LOD score, in other words, and the notation is Z for the LOD score. Let's move on to genome-wide association studies. Simply put, genome-wide association studies are large-scale case-control studies of defined genetic markers. The success of this approach depends on exploiting linkage disequilibrium. As long as a variant responsible for altering disease susceptibility is in linkage disequilibrium, which means they occurs together with one or more of the genotype variants within an LD block, a positive association should be detectable between that disease and the alleles in that block. So it's basically identifying blocks of association with a particular disease. So let's take an example of age-related macular degeneration. So this kind of plot is called a Manhattan plot. So this is a Manhattan plot of genome-wide association study of age-related macular degeneration using approximately 1 million genome-wide single nucleotide polymorphisms, also called SNPs, located along all 22 autosomes on the x-axis. So you can see the blue dots here. They are the ones which were identified to be statistically significant. So there were more of those in the cases compared to controls, and they were actually statistically significant. So these blue dot variants or blue dot genetic loci are considered to be associated with age-related macular degeneration. However, GWAS, or genome-wide association study, does have its limitations in gene discovery, and association does not necessarily equal cause. Just because it's statistically more in the cases compared to controls, there might be confounders, there might be other reasons why it is high. So we can't just say just because there is an association, we have a proof of cause for these particular genes or genetic markers to definitely cause age-related macular degeneration. But if they're statistically significant, we can't completely ignore it. We know that they seem to have some association with age-related macular degeneration. We don't know the biology between the statistically associated variants and the disease, but we know that there is some association. There is also a possibility of artifactual association caused by population stratification, which simply means that in one population we might actually see that these markers are heavily associated, whereas this may not replicate in another population. It may help if ancestry informative markers are used and populations are separated for analysis. We've talked about that in our previous lectures as well. The third way, I haven't put number 3 there, so the third way of also trying to identify disease-associated genes or variants is using genome-wide sequencing. So it could be either whole exome sequencing, where the entire genome is sequenced, but only the coding regions of the entire genome, or using whole genome sequencing, where you sequence the coding as well as non-coding regions of the entire genome. So now you've got, it's very similar to what we do for linkage. So you've got large pedigree, you've identified that there is a monogenic condition, and you've sequenced the variants. But we've got 3 million base pairs in a genome. So we have to then filter out variants that we don't think are associated. So most diseases are caused by variants within the coding region. So let's say we've removed all of the regions which are non-coding. So we've removed about 4 million variants here. So of the 3 billion, we've removed 4 million variants which are not located within or near an exon. So we're left with about 40,000 variants, and we then compare it, look through databases such as gnomAD, for instance. We've talked about gnomAD, which is a database where people with who don't necessarily have any disease, they've sequenced all of their normal genomes, and then if you go and look at these variants, and many softwares can say, hey, these variants are too common in people without any disease. So then we can filter those out, and now we're left with 1,500 variants. And then we look at the functional consequences of these variants. Do these variants actually cause any problem to the gene? And there are many softwares that will help you with that. Sometimes you have to do some of these manually, but softwares can then tell us, has there been any functional consequence to a gene? Sometimes a synonymous variant doesn't necessarily change the amino acid, and therefore there may be no functional consequence. So those ones we can just filter out. And now we're left with about 200 variants, which are, let's say these 200 variants only occur in autosomal recessive disorders. And what we're dealing with is an autosomal dominant disease. So we can then filter out those, and we might be left with 5 to 10 variants. Then we can think about clinical correlation, about look more deeply about what the gene is and see what makes biological sense, and then we might come up with 1 to 2 variants. Even though this flowchart makes it look quite easy, it's not as easy as that to discover genes using whole exome or genome sequencing. We will still need large family trees, and we might still need to combine linkage analysis to the genome-wide sequencing in order to verify. And it's also useful to do international collaboration to understand variants better. There may be other people in other parts of the world who have sequenced variants in a similar way. And if we collaborate with them, and they've also identified a similar variant for a patient with a similar condition, then there is more strength in the variant that you have found. So it's international collaboration and a lot of work in terms of trying to identify these disease-associated genes and variants.